Okay, good morning. So in 2025, the society started the Scott Williamson Award that's going to be awarded annually to recognize achievements and promise in an outstanding early career researcher in modeling and theory in any area of population biology. Scott Williamson was an accomplished early career researcher who worked in theoretical population genetics and evolutionary modeling. He was a humble, kind, and well-liked scientist who made significant theory contributions in understanding the dynamics of natural selection in populations, using a lot of different modeling traditions from coalescent theory to Poisson random fields and sequence substitution models. He applied insights from models and statistics to study selection in lots of different organisms, including Drosophila, humans, rice, and HIV. A native of Kansas, Scott received both his undergraduate and graduate degrees at the University of Kansas. And shortly after starting his faculty position at Cornell University, Scott sadly passed away from brain cancer. The nominations for this award this inaugural year were amazing, and we were so impressed with the quality of the nominees. The winner this year is Dr. Anuraag Bukkuri, who is here today, who's currently a postdoc at the University of Pittsburgh. And we're very happy to announce his award. I'm going to let him talk about his own work, but this is from our award committee. We said that Dr. Bukkuri's creative mathematical approaches have significantly contributed to our understanding of cancer evolution, therapeutic resistance, and the complex interplay between ecological and evolutionary dynamics in biological systems. Congratulations.
Okay, great. Can you guys hear me? Awesome. Well, thank you so much. So my name is Anuraag Bukkuri, a postdoc at the University of Pittsburgh School of Medicine. I'd like to talk to you guys a little bit today on my work on eco-evolutionary dynamics as an engine for cancer treatment and drug discovery. And just as a disclaimer, this talk will probably be more biological and applied than a lot of the other talks that you'll hear in this conference, but hopefully it'll provide some fodder for some nice modeling discussions throughout the week. So first off, I'd like to thank Noah, Joanna, Amelia, and the prize committee, Andy, Katya, and Maria for this award. It's obviously a huge honor to receive this award and to be able to share my work with you guys today. So this is an outline of what I'll talk about. So first I'll give a brief overview of the state of modern cancer research, and I'll talk to you guys about the importance of integrating sort of evolutionary thinking into our approaches in treating cancer. I'll cover a couple of main strategies that we currently have in the field, and then we'll wrap up and we can have some time for discussion. So despite working in a cancer center, these statistics are always still so startling to me. So cancer is really a global health crisis. 10 million people die of cancer each year, which means that every 3 seconds, someone dies from cancer. In addition, it's a massive economic investment. So just last year alone, we spent $200 billion on cancer research and treatment. And this has led to some really awesome advances in radiation therapy. We can zap patients in better ways, right? Chemotherapies, we can give them better drugs, immunotherapies, surgical options. But unfortunately, we haven't moved the needle much when it comes to patient outcomes. And so these are cancer mortality rates in the US for basically the last century, age adjusted. As you can see, we haven't, most of our, outcomes have remained fairly static. We've had some notable decreases, things like lung cancer, stomach cancer, uterine cancers, things like this. But these have mainly been due to public health measures, right? Sort of the knowledge that smoking is bad for you, or improved sanitation, or knowledge of sexually transmitted diseases, rather than anything in sort of, fundamental cancer biology. And so when we put in so much money, so many brilliant people in our field, but we still have poor outcomes, it sort of, perhaps, makes us think that we might have the wrong worldview, right, of how we're going about this. So standard cancer biologists, the way we approach the field is we say, okay, we have a bunch of tumors from patients. We're going to sequence them. We're going to find all of the cancer-associated mutations. And then we'll do these like fancy chemistry, small molecule discovery to target each of these cancer-associated mutations. And we hope that in that process, we will then be able to cure the cancer. Unfortunately, this doesn't work too well. So this is, therapeutic resistance is the main reason of treatment failure in advanced cancers. So this is unfortunately all too common in the clinic. So a patient presents here with this aggressive form of melanoma, a skin cancer, and he's given a BRAF inhibitor for 15 weeks. So this is a targeted treatment, again, going after the specific oncogene BRAF in the cancer. 15 weeks later, the physician says he's all cured, he's sent home, but then just 8 weeks after that, the disease is back and it's worse than ever. And at this point in the clinic, there's nothing that the physician can do, right? So the patient is given palliative care and then he passes away soon after. And this is basically the most common case for cancer patients today. And so, I think, what we've learned from Jurassic Park is that really life always finds a way, right? So no matter how smart we are, no matter how many drugs we come up with, life will always find a way. And so I thought I sort of want to transition our thinking about cancer as a disease of the genes to thinking of cancer rather as a disease of evolution. And so how can evolutionary principles help us treat cancer? So the first thing to recognize is that as soon as we treat cancer, right, as soon as the physician starts administering therapy to cancer, we are engaging in an evolutionary game against the tumor. So if you do not kill all of the cancer cells, the tumor will respond in ways that ecologically and evolutionarily compromise patient health. However, there is one key advantage that we have against the cancer. So cancer cells are entirely teleonomic. They have no anticipatory ability. They can only respond to what's happening to them in the moment. However, physicians, if they're smart, can be teleologic, right? So they can anticipate and steer the eco-evolutionary dynamics of cancer into desired phenotypes. And so current cancer treatment paradigms, unfortunately, forfeit this advantage. So what happens when you go into the clinic? Generally, the standard of care is physicians will give you a maximum tolerable dose, or MTD. So they'll give you the therapy for as long and as hard as possible. Right? And so what happens is you might start out with this tumor here on the left. So it'll primarily be composed of treatment-sensitive cells. Those are the blue cells. You might have a few of these treatment-resistant cells. And so when the physician gives this maximum tolerable dose, it'll be extremely effective at decreasing the population size. And so here, this is sort of a scan of a patient with lung cancer. So the gray regions are the regions that are clear, and then the light gray regions are the regions where you have this cancer infiltrate. So when you give that maximum tolerable dose, you are going to clear up the tumor. Right? So this is good. But unfortunately, what you're inadvertently doing is you're specifically selecting for the resistant cells, right? So you're getting rid of all the sensitive cells, and the resistant cells will be the only ones left over. And as you continue giving this maximum tolerable dose, eventually you'll have only resistant cells, you'll have this outgrowth of resistant cells, and then this is what we call sort of, treatment resistance, recurrence, relapse, and the cancer is back. And again, at this point, this therapy no longer works for the patient. So if there is another therapy, we might give that again and start this process over again. But more often than not, this is, again, the end of the road for the cancer patient. And so one way that we might combat this is using this idea of adaptive therapy, which uses principles of competitive release from ecology to sort of delay the progression of resistance. So these models usually have three key assumptions. So they often assume pre-existing resistance. Some of our work has shown that this assumption can be relaxed. We also assume that resistant cells outcompete sensitive cells under therapy, right? This is a bit obvious. Resistant cells don't die from the therapy as much as sensitive cells. But then we also often assume a cost of resistance. So under baseline conditions, when there's no therapy, sensitive cells can outcompete the resistant cells because it's usually more metabolically costly, for example, to evolve these resistance mechanisms. And oftentimes when we're developing mathematical models, we usually sort of just have this based on sort of standard logical-Volterra competition equations. And we can then go and try to simulate various treatment protocols or optimize these on a per patient basis. But the main idea here is that you can start with the same tumor, right? Primarily consisting of those sensitive cells. You can give therapy to debulk the tumor, but you intentionally leave some sensitive cells around. And what that does is that when you remove the therapy, it can then suppress the outgrowth of those resistant cells. So you let the tumor grow back, and then you give therapy again. You let the tumor grow back, you give therapy again. And so in this way, you can sort of control the tumor for a much longer period of time. And so we actually applied this in a phase 1 clinical trial at Moffitt for castrate-resistant metastatic prostate cancer. We did this the protocol was a fairly naive way, so we didn't even have any sort of optimization. So we just administered the drug until the tumor burden dropped by 50%, and then we stopped therapy. We waited for the tumor to grow back, and then we re-administered it when the tumor is back to its original size. And so you can see the outcomes here. So these are called Kaplan-Meier curves, where you're plotting sort of the estimated survival of this population over time. And this blue curve represents the control patients that just received standard maximum tolerable dose. And this red curve represents our adaptive therapy. As you can see, we had an average median improvement in overall survival by about 20 months. And just to give you a sense, the average FDA-approved anti-cancer therapy prolongs life by about 2 months. So achieved a 10-fold increase in survival just by scheduling these drugs in a smarter fashion. And again, we didn't even optimize any of the math here, right? So there's a lot more improvement that can be done. In addition, we provided the patients with an average saving of $70,000 a year. And whether you know it or not, actually one of the main barriers in cancer treatment in the clinic is that patients actually won't take the therapies if they have to pay more than $3,000 out of pocket, right? Over half of old patients don't. So in this case, patients will take our therapies and they will live for much longer. There are a lot more open questions here, so I definitely love to talk to people who are interested in this, over the week. But we still don't know necessarily what are the necessary components for mathematical models of adaptive therapy, both broadly and in particular cancers. We still don't know when we should be aiming for cure versus this long-term control, right? How can you model this? You could, integrating optimal control theory to figure out what are the optimal dosing schedules on a per patient basis, and also integrating this closer with experimental results and clinical practice to sort of improve translation. So a lot of open questions here. So when I was working on some of this adaptive therapy, I actually spent some time as a science writer. I was Miami Herald science writer for a summer, and I covered this particular camp. So this is a camp for kids with cancer, right? And so we have around 20 kids that come every year to this camp, and usually at least 3 or 4 of these patients will die during the camp, right? So this is sort of their last time to just have fun and not be tied to a hospital bed, right? And this was, I think, really impactful for me and really, impacted, the course of my future research, because this lady right here, Dr. Pefkuru, who ran this, said that these kids leave with the belief that they're going to be healthy one day and that they're going to enjoy life like everybody else. And so it's made me think, okay, it's great, if we can extend therapy with using this adaptive therapy, control the cancer for a couple more years. But unfortunately, this is not good enough, right, for many of our patients, right? And so this leads to the idea: is there any hope left for cancer eradication? So in the field, we've sort of gotten away from this idea a little bit, but now we're I want to think, is there any way that we can still cure cancer? And so this led to the idea of sort of evolutionary traps and double binds. So this is often used in ecology. So for example, if you're a gerbil here on the Negev desert, you have 2 main predators. You have snakes and you have owls. So you might try to hide from the owls by sort of going into these small burrows in the desert, but that's where the snakes lay hiding and trying to get you. Or you might be trying to hide away from the snakes by skirting out into the open, but that's when the owl can come get you, right? Yes, this is produced by ChatGPT. That's why the snake looks a little weird. Right? So this is sort of what we want to do. Nice little donut snake for you, right? Yeah. So this is sort of what we want to do with cancer, right? We want to administer one therapy to strategically shift the selection pressures in this tumor to push this gerbil or this cancer into a state that's more easily targetable by the second therapy. So sort of set this evolutionary trap. And so we've done this in a variety of ways. So these are some of our examples. So they actually showed that providing excess glucose or excess nutrients to the tumor actually promotes this tragedy of the common. So it makes these cancer cells very competitive. It also promotes this invasion of defector phenotypes, so cells that don't produce growth factors and sort of free ride on the public goods being produced by other cancer cells, which then makes cells vulnerable to GLUT1 inhibitors or starvation therapy. Similarly, one of the main problems in cancer therapy is this idea of heterogeneity. So now we're thinking of drawing on principles of coexistence theory to understand the mechanisms of coexistence of various cancer clones, and then we try to administer one therapy that modifies the microenvironment to homogenize the population, and the second therapy can then more easily eradicate the population. So looking at this in the context of breast cancer and bladder cancer, and then we'll talk to you guys about today, where we're looking at how therapy can actually induce what's called a whole genome duplication. So cells can duplicate their DNA without dividing, but then that makes cells vulnerable to death via these endocycle targeting therapies. And I'll talk a little bit more about what that is in a minute. So to really design these evolutionary traps, we need to understand how cells respond to therapy. And so when you give a population of cancer cells this drug, there are 2 main ways that these cells can adapt. So one is via evolution. So this is just standard genetic evolution, heritable changes passed down from generation to generation. These are sort of the standard mutations that you hear about. But cells can also acclimate to the stressor, right? So they can undergo behavioral changes in their own generation. They can migrate to a safer habitat, right? They can enter a dormant state. They can sort of go to sleep and escape this therapy. So these are the couple of ways that they can respond. So sort of thinking about how we can understand evolution, there are many ways we can do this. One way is using this G-function approach. This just consists of a set of coupled ODEs. So we have the population or ecological dynamics given by this equation here. So a change in the population size over time, dx dt, is given as a product of the current population size x. and this G function, which is sort of the fitness generating function that captures the per capita growth rate of these cells as a function of their evolutionary strategy V. So this could be like the number of antipores on a cell surface, and then X, the current population size. And then the trait or evolutionary dynamics sort of given by Fisher's fundamental theorem here, the change in the strategy over time given by a product of evolvability, the capacity for a population to generate heritable variation that can then be acted upon by natural selection, and the selection gradient here, dg/dv, which is just saying if you perturb your strategy a little bit, how does that impact your fitness? And so oftentimes this is visualized on an adaptive landscape. These adaptive landscapes are usually changed through time as the density and frequency of these various morphs also change. But here we sort of presented this sort of a static landscape. Okay. So basically you can plot the fitness as a function of trait value. And so if you're a cancer cell at this yellow circle here, you'll try to climb this adaptive landscape to reach a local peak given by a screen start A. And the rate at which you do this will be dependent again on your evolvability or your step size, and then the slope of this adaptive landscape, or how extreme the selection pressure is. So the more you're under stress, the higher your evolvability, the faster you're going to evolve. So this is sort of well known, but then the issue comes when you want to incorporate acclimatization, which is a bit trickier. So how do cells acclimate? There are many ways they can do this. What we'll be thinking about here is them doing so by entering a different cell state. So for example, they can become more epithelial or more mesenchymal, right? They can become more squishy, helps them be more motile, find resources more easily. But the idea is that including plasticity requires that we take this life history or demographic perspective, which leads to this idea of state structured populations. So what do I mean by this? So for example, if you saw a butterfly and a caterpillar and you didn't know any better, you might treat them and model them as completely separate species. And you actually might be fine doing that in most scenarios. But unfortunately, as soon as you consider the underlying evolutionary dynamics of the system, you'd run into problems because what happens in the caterpillar stage impacts what happens in the butterfly stage and vice versa. And so the idea here is that we need some sort of unifying fitness function that allows us to track fitness across all these states in the life cycle of the butterfly. And one way we do this is using the state structure modeling approach. So we initially did this in the context of baboon reproduction, which is a really interesting sell to folks in the Cancer Center for why I should be able to work on this. But we did this a little incognito. But we basically start from a life cycle graph that, for example, can capture transitions between young, reproductive, and old baboons and how these dynamics are. Or you can start from your set of equations. You can encode all that information into this matrix, right? And this matrix is called this population projection matrix. So if you're in Maria's awesome discussion yesterday, we talked a lot about this. And so you can take this population projection matrix, and the trick here is you can use the spectral radius or bound of that matrix as the G function because that controls the long-term dynamics of this population. And so a lot more details behind this that are described in some of these papers, but we've taken this framework and extended this to provide sort of stochastic analogs to look at, what happens if you have fluctuating non-constant environments, what happens if you have continuously structured environments or environments structured in multiple ways, how are there sort of analytical techniques and tricks that you can use to analyze these populations. But this is the main idea for how we're going to model these situations. And considering the state structure is especially important in the context of cancer, because as we showed in some of our recent work, cancers really demonstrate increased cell cycle plasticity. So they can go through the cell cycle in a bunch of different ways. So these are called cell cycle maps, a sort of technique that our lab is sort of working on now. And so you don't really need to understand what exactly all these mean. These are just different ways that the cells are traversing through the cell cycle projected on sort of a two-dimensional landscape here. And so all of these are breast cancer cells, except this. This guy is just a normal breast cell line. And as you can see, there's just a lot more complexity going on in these cancer cell lines. There's a lot more ways they can go through the cell cycle, which is a big problem for therapy because that means there are many ways that they can actually evade the effects of therapy. In addition, we showed that cell cycle plasticity can really play a key role in all aspects of cancer, from tumor initiation to progression and resistance. And so the key idea here is that we want to target cell cycle plasticity because doing so can actually constrain the pathways to evolutionary rescue, which is what gives rise to therapy resistance and recurrence. So most people in this field are just focusing on what happens after you get the mutations, what are other therapies that can work to combat that. Here we want to stop it even before it gets there. We want to prevent the evolutionary rescue in the first place, right? And so what we'll be looking at here is a case study of how this works. We've done this in various cases, but this is one that is particularly interesting to me. This is called the polyaneuploid cancer cell, or PAC state. So what does that mean? Polyaneuploid composed of two words, polyploid and aneuploid. Polyploidy referring to whole genome duplication of a baseline genome. And that genome oftentimes in the context of cancer is aneuploid. Right? There are a lot of chromosomal gains and losses in various parts of the chromosome. And what this is characterized by is a shift from the canonical mitotic cycle, where you go from G1, S, G2 to M, to this non-proliferative endocycle, in which the cell undergoes repeated G and S phases without ever entering mitosis. So what happens is you get this increase in genomic content without cell division. The cell gets really big. It has a lot of DNA. And what's really scary about this state is that it forms in a condition-dependent manner. And so, for example, here we have prostate cancer cells, PC3 cells, that are just at baseline. And these cells primarily exist in that diploid 2N state. But when you administer chemotherapy, you see cells rapidly transition into this PAX state. All the non-PAX die off. Right? So this is really how the cells arrive. This is what's promoting this drug resilience. And then when you remove the therapy, the cells can recover back to their pre-therapy frequencies and counts, and you sort of recover a similar sort of morphology as well. And you can see that clearly here. These are the same cells exposed to chemotherapy, and you can appreciate just how large these cells are getting over time as they undergo this therapy-induced endocycle. Couple more points. So cells in the PAX state are non-proliferative. So you can't divide when you're in the PAX state. You can only depolyploidize to the 2N cells. And so here we took these PC3 cells to investigate this. We injected them with a dye. And the idea is if they're proliferating, the dye will be rapidly diluted. So these are the standard prostate cancer cells, and these are the PAX. So these cells rapidly dilute the dye, whereas in the PAX, it remains fairly stable. You notice this decrease in intensity because the cells are getting bigger, but it does remain stable. In addition, we've taken PAX and injected them into mice. And what you can see is over 15 weeks, the tumor hasn't grown much. But via bioluminescence imaging, we can see that they're metabolically active. So they're doing things, but they're not dividing. Another thing that's really scary about these cells is we took these prostate cancer cells, we gave chemotherapy, generated those PAX, isolated them, and exposed them to various therapies at very high concentrations. And you can see that their viability is 100% really. Right? So they don't die from therapy. So these are entirely therapy resistant. So you can start to see why these cells, again, are so scary and why they really see this evolutionary rescue that then goes on to lead to therapeutic resistance. So with this, we can create sort of a model of PAX. So again, just as a refresher, this is our life cycle graph. So the 2N cells can either divide or transition into the PAX state. The PAX can't divide. They can only depolyploidize into the 2N state. PAX are therapy resistant. The 2N cells are therapy sensitive. And so we assume that the 2N cells grow in this logistic manner. And then we have these blue terms, which capture the obligate or background transition. So just at baseline, these cells are transitioning between these states at some rate. And then we have these red and pink terms, which are the drug-associated terms. So again, we assume that cells in the 2N state die from the therapy in sort of this Michaelis-Menten form. And we also have these facultative or stress-induced transitions. So all this is saying is that the more stressed the cells are, the more they're going to transition into that PAX state. And then we have our evolutionary dynamics like we described before, and that comes in our drug-associated terms. So as you evolve resistance, you're drug-induced, you die less from the therapy, and you transition to the PAX state less frequently. And so with this model, we then looked at various ways that we hypothesized that the PAX state can lead to therapeutic resistance in the strong inference approach. I'll just present one that was in accordance with some of the experimental data that we're seeing, this idea of evolutionary triage, in which we assume that evolvability is dependent on the frequency of PAX in the population. Why is that? Again, because PAX have this increased genomic material, they have more room for evolutionary experimentation. And so here we've just simulated this, very basic simulation. So we have the top panel capturing the population dynamics of the 2N cells in PAX in blue and red, and you have these drug resistance dynamics on the bottom. Regions in white are regions without therapy, and this region in gray is the region with therapy. And so before therapy, you see that this population primarily exists in the 2N state. This is what we see in our cells, in culture, in mice, and patients. When you administer therapy, you see that 2N population really comes crashing down, and you see that rapid transition to cells in the PAX state. As the cells recover and evolve resistance, that PAX population gradually starts decreasing, and you see this recovery in the 2N population. You remove therapy, they revert back to their pre-therapy frequencies and counts. There's also a lot more going on behind the scenes that I'm not presenting here, but if you're curious, these cells do a lot more scary things, so feel free to ask me about that later. But the key point here is that just from the models, just from our eco-evolutionary models, suggested this idea of life history and measures to combat resistance. So this is an entirely new class of therapeutic strategies in cancer. That's, I would say, a subclass of evolutionary double binds in which you administer chemotherapy in conjunction with PAX-targeted therapies. So how does that function as a double bind? You administer chemotherapy, you push cells into that PAX state. But if you target that transition, you can prevent cells from entering that state, force them to go into mitosis, but then because the cells have so much DNA damage, they won't be able to complete that successfully, and they'll undergo mitotic catastrophe and die. Right? So that was the idea. That's great in theory. Does it work in the real world? So again, I'm covering up a lot of molecular biology here, but we basically found, hypothesized that these two key proteins, HIF2 and AP1, may be important in mediating this pack transition. And so again, using exactly the theories that we hypothesized with our modeling, we did this experimentally, and we showed that in all cases across a variety of cell lines, combining our chemotherapy with this pack therapy leads to a significant reduction in tumor burden. At this point, you may be saying, great, that's cool, but you still haven't eradicated the tumor, right? As long as you have these cells, you can still get, evolutionary rescue. And this is a good point. We sort of did this in a rather ad hoc way. So then the next question we asked is there a systematic or data-driven way to study the mechanisms of polyploidization? And this is what we're currently working on. So first, we try to understand the temporal dynamics of polyploidization. So when does this actually happen? So we can take cells from mice or patients. We can engineer them with cell cycle biosensors. What that means is that the cells will glow different colors depending on which stage of the cell cycle they're in. We can culture them in normal media or disease media that we've made to sort of recapitulate some of these liver diseases, this accumulation of fatty acids, or therapy. And then we can perform time-lapse imaging. So we can follow these cells over time to see what they're doing. And we can use deep learning to segment and track cells at an individual level to see what's going on. Right? So with this, we can understand what are the temporal dynamics of polyploidization, and then we can look at what are the molecular mechanisms. So this is a pipeline that we're developing now. So we can start with these cells or tissues from patients, and we can stain them with antibodies. And so critically, we can get really up to 50 different antibodies here, and we can stain all of the cells so we can get subcellular resolution, quantification of each of these protein levels in the nucleus and cytoplasm, and from that we can extract all the features here, and we get this very large data matrix of all the cells and the expression levels of all of these proteins. And then we can use some computational algorithms to generate these cell cycle maps. I'll talk about what this is in a minute, but this will then be used to inform our endocycle targeting therapies, right? And so this is just how it looks. So these are tissue samples, all from the same patient, all from the same cells. And you can again appreciate these are stained with multiple different antibodies. And you can very clearly see that you can really get the subcellular readouts of protein levels in each cell. So from this, again, this is our cell cycle map that we've created. And we've clustered cells here. So they start in this purple G1 and go into this green S phase, down into this blue G2. And then they can either undergo canonical mitosis up into this red M phase, back into the cell cycle, or they can exit the cell cycle and undergo this endocycle here in orange, right? And we can also overlay various proteins onto the cell cycle map. So these are some of the key ones. For example, RAID, this is involved in the management of supernumerate chromosomes and DNA damage. So as cells enter the endocycle here, so again, all these graphs and this, just the geometry is the exact same. The color schemes are different. So here in these four graphs, the higher, the warmer the colors, the higher sort of the concentration of the protein. So as cells enter the endocycle, again, they have a lot of DNA damage, which is why this is a stress response. As they resolve that DNA damage and go back into a cell cycle, the DNA damage is resolved. Another key thing, cool, CDK1 is the canonical mitotic CDK. You can see that as cells go through that mitotic trajectory, they increase CDK1 as expected. But critically, these are some of the key proteins that we've identified. So WEE1 seems to mediate that transition. So we have a sort of WEE1 and P53 mediated inhibition of CDK1 and a switch to CDK2 that allows us endocyte. So now in a very data-driven way, we now know what are the key proteins that mediate this endocycle. So then we can go in and target each of these proteins and see what happens. If we target these proteins, can we then shift cells, block this trajectory altogether, and force cells to undergo mitosis, in which case they'll die from mitotic catastrophe. And so this is some of the current work that we're trying to do today. So the conclusions, I'll just wrap up here. I think ecological and evolutionary thinking is really critical in modern cancer research. I think a lot of the ideas that we've heard and will hear in this conference will be really applicable to a lot of questions in trying to address various aspects of cancer, especially therapeutic resistance. This new idea of sort of life history and line therapies may provide paths to cancer eradication by removing these sources of evolutionary rescue, so not allowing cells to even evolve resistance in the first place. And I think integrating mathematical, computational, and experimental approaches can really provide rigorous, systematic approaches to drug discovery. And so with that, I'll just thank our lab and our funding sources, and I'm happy to take any questions.
We have 3 minutes for questions.
Yes.
So in your intro, you gave two motivating examples, prostate cancer and childhood cancers. But I think the ecology of childhood cancers is really different from those of cancer aging. have you thought about the implications of that for these evolutionary treatments?
Yeah, it's a good question. So
Repeat it for the people in the back.
yeah, so Fred was just asking about, the ecology of childhood cancers being very different from the ecology of standard adult cancers, things like prostate cancer, and so how that might impact some of our evolutionary therapies. So, I mean, a couple points. One is the extinction therapies that we're trying to do with these evolutionary traps are not just applicable to childhood cancer. So that was more of just a personal motivation for going into that field. So this is one point. But yeah, I mean, you're definitely right that childhood cancers have very different dynamics. And sort of the thing that we're working on with like cancer corruption and criminology is really trying to get at that aspect because I mean, as we've discussed before, right, in a lot of developing societies, cellular societies and whatnot, the controls on sort of, keeping control of cancer or cheaters are actually diminished. And that's to the benefit of the society, whether it's a human society or a cellular society, because it sort of greases the wheels and gets things going. But then as the cancer sort of develops, this is when those control measures start to get put into place and corruption levels decrease. And so what we've seen a lot of childhood cancer. So for example, in neuroblastoma, we see some of these really counterintuitive observations where you might have a stage 4 neuroblastoma. Right? So you might have a kid that comes in, the doctor says, this is stage 4, this is metastatic, there's basically no chance of survival. And the physician will not even administer any therapy, but somehow magically the tumor will go away on its own. Right? So you get spontaneous regression of this tumor, and we still have no idea why. And so some of these more, I think, social science perspectives can really, I think, shed light on that rather than just traditional sort of evolutionary approaches. So, yeah. Yes.
Sort of similar question about whether spatial structure affects the dynamics of competition in ways that would, lead to different sorts of models than the ones you present.
Yes, so definitely. So I didn't present any of the spatial stuff, but we do have work on looking at like habitat structure populations. So what happens when you have various habitats. And then we're also now developing this idea of cancer community ecology. And the idea is that based on the spatial structure of where cells are in the tumor, you have different life histories. So for example, cells that are near the outside of the tumor, right, will have very different selective pressures. So they have a lot of nutrients, but also a lot of immune sort of infiltrates, right? Whereas cells in the middle won't have as much nutrients, but also won't have that immune pressure. And so this leads to very different dynamics. And so then we can think about these same evolutionary traps and say, okay, if we give one therapy that targets that outer ring, something like chemotherapy, right, that can get rid of these rapidly dividing cells, and then we administer another therapy that's specifically like an immunotherapy that specifically targets those inner cells, that might lead to better outcomes. Right? And so we're thinking about ways to take advantage of that. Yes.
Have you found in your work any use of analogies from like the antibiotic resistance literature and kind of scheduling of drugs in the antibiotic resistance?
Yeah, no, it's really great that you asked. Like 2 weeks ago, I was running helping run this Evolutionary Medicine Summer Institute in which we specifically looked at the parallels between cancer, antibiotic resistance, and pest management. And so all of these are very intertwined. We have one paper looking specifically at comparing cancer evolution, models of cancer evolution and bacterial evolution. So there's a lot of parallels. I think we'll hear some other talks in this session that'll touch on that as well. But ideas of biofilms are very closely related to this ideas of PACs, these state structured populations. Some of the more intricate dynamics are different. For example, adaptive therapy isn't considered so much in the bacterial world. You sort of want to get rid of the bacteria. But there are definitely a lot of parallels that we're investigating. Yeah.